Have you ever stopped to wonder what actually happens when biology generates way more data than human beings could ever possibly read? For centuries, biology was pretty much an observational science. You look through a microscope, you jot down notes on what you see, and that's that. But today, biological science is producing an insurmountable, literally exponential amount of information. We've hit this wild tipping point where traditional, manual observation just absolutely cannot keep up with the sheer volume of genetic data pouring out of labs all over the world. Take a look at this number, 150 billion. Let that sink in for a second. As of 2013, that was the staggering number of DNA bases stored in GenBank. If you aren't familiar, GenBank is the NIH's genetic sequence database built by the National Center for Biotechnology Information. It is packed with publicly available DNA sequences covering the genomes of over 165,000 different organisms. We're talking everything from elephants to apple trees, humans to microscopic bacteria. It really puts into perspective the massive, almost unbelievable data explosion problem we're facing right now.
So, welcome to The Explainer. Today, our topic is bioinformatics, decoding the biological machine. We're going to dive right into how bioinformatics serves as the ultimate game changer for managing this ocean of data. It's the vital discipline that actually allows us to translate the literal code of life by tapping into the incredible power of modern computation.
Here is a super quick roadmap of where we're headed today. We'll start with the genetic data explosion, then look inside the biological machine, track the history of the Human Genome Project, really define bioinformatics, and finally, look at unlocking biological mysteries.
All right, let's jump right into part one, the genetic data explosion.
Get this. Submissions to GenBank are currently pulling in over 3 to 4 million new DNA sequences every single month. That is a literal data avalanche. It represents an exponential growth rate of sequencing data that just hasn't slowed down at all since the early genome projects wrapped up. And it makes sense, right? It's fueled by our intense drive to understand biological diversity and, of course, prevent disease. But this relentless, nonstop influx of data forced biology to turn to computer science just to make sense of the whole shebang.
Which brings us to part two, inside the biological machine.
So, if we zoom way into a typical animal cell, it's basically a bustling microscopic city. Every cell is packed with hundreds of different macromolecules. You've got the mitochondria, those little oval power plants oxidizing carbohydrates for energy. You've got the folded, winding structures of the rough endoplasmic reticulum carrying ribosomes to build proteins. But the true vault, the absolute command center, that lies right in the center, the nucleus, and it carries the cell's master genetic blueprint in the form of DNA.
The crucial thing here is understanding what exactly is locked inside that nuclear vault. That is the genome. The genome is the complete genetic material of an organism, fully encoded in its DNA. Now, you probably know about genes, those discrete protein coding units. But here's the kicker. A genome isn't just genes. It encompasses those functional genes, plus the vast, sprawling stretches of non-coding sequences spread across all your chromosomes. Trying to map and understand this entire massive package, not just isolated genes, is the monumental task of modern biology.
Moving on to part three, the Human Genome Project. Let's trace how we got here. Biomolecular sequencing actually kicked off way back in 1951 when scientists Sanger and Tuppy figured out a 30-residue protein from insulin. By 1982, we finally saw the very first release of GenBank, but the real explosion? That hit in 1990 with the launch of the Human Genome Project. This was a massive 13-year global effort to map all 3 billion chemical base pairs of human DNA, and they officially completed it in 2003, perfectly timing the big announcement with the 50th anniversary of Watson and Crick discovering the DNA double helix. Talk about a milestone.
Now, here is a fantastic reality check. When mapping the human genome, scientists totally assumed we'd have something like 100,000 genes to account for, how highly advanced and complex we are. But the shocker? The human genome only contains about 25,000 genes. Compare that to the simple C. elegans moundworm, which sits at approximately 20,000 genes. That's right, we humans barely have more genes than a microscopic worm. It was a total wake-up call for the scientific community. It proved definitively that biological complexity is not just about a simple head count of genes, it's all about the complex, intricate regulation of those genetic networks.
Okay, part 4, defining bioinformatics.
So, how do we solve this mind-boggling data problem? The answer is bioinformatics. Think of it as the ultimate translator between two completely different worlds.
On one side, biology speaks a descriptive and experimental language. It's all about observation, messy lab tests, trial and error. On the other side, computer science speaks an analytical and precise language. Algorithms, solid math, rigid data structures. Bioinformatics builds a bridge right over that gap. It allows us to take the hyper-precise algorithmic problem-solving of computer science and aim it directly at the deeply complex, messy experimental data of living systems.
And that leads us beautifully into Part 5, Unlocking Biological Mysteries.
When these computational models try to build out massive evolutionary family trees, they rely on parsing a really critical distinction: orthologs versus paralogs. They are both homologous, meaning they share an evolutionary history, but they split differently. Orthologs diverge because of a speciation event. For example, the ANTP gene in fruit flies and the MAV5 gene in roundworms evolving from the exact same common ancestor. Paralogs, though, diverge via a gene duplication event within the ancient ancestor itself. We absolutely need these powerful algorithms to sift through billions of data points just to make these distinctions, revealing exactly how nature reuses its most successful patterns over millions of years.
The real-world applications happening right now are just mind-blowing. Take protein folding. A linear chain of polypeptides folds into a 3D structure in literal seconds. Bioinformatics tries to computationally predict this because that final 3D shape completely dictates what the protein actually does. We use sequence alignment to find hidden similarities between species. We use microarrays, these tiny gene chips, to monitor thousands of genes all at once, spotting specific cancer mutations in record time. And we map out gene regulatory networks to decode the essential on-off switches of the cell. These tools aren't just helping the physical lab, they are totally revolutionizing it.
There's a quote that I think absolutely hits the nail on the head here. In silico life science research translates raw experimental data into workable models or simulations to identify targets for drug development. Now, in silico literally just means in silicon, as in on a computer chip. With these tools, we can literally simulate and model life digitally. We can identify potential therapeutic targets and test complex biochemical pathways on a screen, long before a scientist ever even has to pick up a pipette in the lab.
So as we continue to navigate this incredible post-genome informatics era, I want to leave you with a question to really chew on. How will these integrative computational models ultimately unlock the future of personalized medicine? Because the data's already out there, the computational power is growing exponentially every day, and very, very soon, your highly unique individual genetic makeup could be modeled in silico, giving doctors the ability to provide medical care that is specifically, perfectly designed just for you. Pretty amazing stuff, right? Thanks for joining me on this explainer.